In this video, we will discuss about a summary of whatever we have discussed in our several videos. That is glycosylation on the pancreatic cancer and its impact on the immune response. So we will start with this first point, that is glycan elongation, that prevents the immune cells to come and interact with the cancer cells. So what happens in these cancer cells, because of the mutations, their elongated glycan structures are formed, which will also cause the increase in the thickness of glycocalyx, and this will leads to create a steric hindrance that will prevent the various immune cells such as T cells, natural killer cells, dendritic cells, and macrophages to come and interact with the uh, cancer cells or to the non-self epitope that are expressed on the cancer cells. And since there is no interactions between immune cells and the cancer cells or their non-self epitope, they will not be able to generate any immune response, since the immune cells will not detect any harmful substance within the host. So next part, so this is just a first step, first perspective, how these glycans prevent the T cells to come and interact with the cancer cells and generate immune response. And next aspect of this dysregulations of the glycans within the pancreatic cancer is about increased silations that will cause monocyte differentiations into immunosuppressive macrophages. So in this case, what happens is that because of the reprogramming in the pancreatic cancer cells, this leads to the upregulation of the enzymes such as ST3-GAL1 and ST3-GAL4, and these enzymes are responsible for sialic acid. So the upregulations of this enzyme will cause upregulations of the sialic acids on the pancreatic cancers, and these sialic acids are responsible for directly interacting with the Siglec-7 and Siglec-9 that are expressed on the monocytes, and thereby helps in differentiation of this monocyte towards M2 phenotype macrophages. And this M2 phenotype macrophages has the tumor suppression properties, and they act like a tumor associated macrophages. And we have discussed this thing in our video that monocyte can be differentiated into two phenotype. First one is M1 phenotype that has the tumor suppression properties, and next one, the other one is this M2 phenotype that has the tumor progression properties. So in this case, the monocyte will differentiate into this tumor associated macrophages or M2 phenotype that has the tumor progression properties by the sialic acids that are overexpressed on the pancreatic cancer cells, interactions with the Siglec-7 and Siglec-9 that are present on the monocytes. So this is the second perspective, and the next one is this mucin derived pro-inflammatory secretions. So what happens in this case is that there is upregulations of the mucin 1 within the this pancreatic cancer cells, and that will cause a release of this inflammatory cytokines. And these inflammatory cytokines can interact with this or interact or leads to the generation of these regulatory T cells and these myeloid derived suppressor cells, and both of these cells has the functions to control the immune activations. In general terms, this regulatory T cells functions is to prevent the autoimmune response by preventing the cells to attack its own cell, by preventing the immune cells to attack its own cells. However, this property of the regulatory T cells will be exploited by the cancer cells through these upregulations of this mucin 1 that will lead to the inflammatory cytokines, and uh, that will cause these regulatory T cells to suppress the immune response, and thereby it will this whole upregulation of the mucin will cause the suppression of the immune response against the pancreatic cancer cells. The last one is about the inflammatory cytokines has the reverse impact or regulates the glycosyl transferase. So in this case, what happens is that inflammatory cytokines in the tumor microenvironment will cause the upregulation of these enzymes such as ST3-GAL3 and ST3-GAL4, and also this FUT1 to FUT2 to 6, FUT6, okay. And this upregulations of these enzymes will lead to this aberrant sialic acids structures formations, which are commonly expressed in the pancreatic cancer cells, such as Lewis X, S Lewis X, and S level Y structures. And these structures thereby will these structures will thereby helps in the immune suppressions, or either through the ways that is mentioned in this step 2, or they will also cause act as a this steric hindrance manner, and prevent and act as a physical barrier to prevent the immune cells to come and directly contact or interact with these pancreatic cancer cells. So these are some of the ways by which this dysregulation in the glycans leads to the suppression of the immune response, and at the same time it will promote the tumor progression. I hope you got a basic idea about how this basic idea and the whole summary about how the glycosylation on the pancreatic cancer has an impact on suppressing the immune response. We have discussed individual parts uh, in our previous videos. If you need to understand or go through the individual parts, you can either refer to the previous videos, or you can also go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.